Okay, so hi everyone. Yeah, Joseph Geraci. Just for background, I'm a mathematician and a computer scientist and a medical scientist. And I'm excited to be here and to talk about this project with Jay. So basically, we're one of the only companies to build technology that actually sits in between the data that we want to analyze and the machine intelligence that people want to utilize. We built this actually to help pharmaceutical companies through their clinical trials. We're trying to shorten trials by understanding patient populations in a much deeper way, and that's what I'm going to talk about today. And we also do some drug discovery. I'll touch upon that briefly. So what we're trying to do is we're trying to deal with a situation where these data sets are small. So a lot of really powerful methods, like, many people have been talking about deep learning, these are very powerful methods that replicate very well, but you need massive amounts of data. That's one problem. The other problem here that we have with Jay's project is that, and with pharmaceutical companies in general, is that a label like schizophrenia versus control that Jay's coming to me with, or Alzheimer's versus control, that Alzheimer's label is very loaded, okay? And there's a lot of reasons for someone to enter into an Alzheimer's state. And so what we did is we created a technology that literally shatters and stratifies the patient population so that machine learning can do a better job, right? So that's the way to think of us. We empower artificial intelligence to do the type of stuff that Jay and pharma companies want to do. And in this case, it's understanding what is driving the disease. So on that note, if you look here on the screen, these points that are glowing, right, sending out sonar signals, these are variables. And what we do is we're really focused on causality. And this is a very big problem because when you have hundreds or thousands of variables, like in our case, it becomes a very, very bad problem to find what's optimally driving disease. And in this case, right, to help Dr. Anna figure out what role is mitochondria really playing, this is where a tool like this becomes critical, because in this technology, the variables can hear each other at a distance. And so this kind of shortcuts the massive amount of combinations that otherwise we'd have to use. And so then what ends up happening is, because we get these different causal perspectives, what we end up getting is this mosaic of theories. And so each of these panels you're seeing have dots sprinkled inside, and each dot is a patient. And what happens is each of these panels becomes a hypothesis about a subpopulation of people, right? Saying, hey, this is what's driving this subpopulation. Does this make sense to you? And that's what Jay was referring to as augmented intelligence. And so here what you're seeing is actually somebody using this technology for this amazing ALS competition, this global competition that I was actually a judge in, and then I participated after I judged the winners. And so what we did here essentially, so let me get into here and explain to you what's going on here. So what you're seeing here is a map of ALS patients, and each of these points is a person. So this person's hovering over this patient or their patient sample and showing that there's these genes associated with the patients in certain levels. And so this allows a user like Jay to actually understand how the machine honed in on a certain perspective. And what you're seeing here is the ability for the machine to actually explain, why are these 13 people separate from everybody else? And the amazing thing is we've been recognized as being one of the only companies to be able to actually shatter this data set. That's what we call shattering. But really what this is it's a landscape of how the patients relate to each other. That's what it is. And what's interesting is the blue patients are the patients that have ALS initiate in their limbs, and the red group that this person's zooming in on. So you can see the blue people moving away. There's less and less difference, right, between the limbic. So those first 13 people had a major difference. And look what this person uncovered. This amazing subpopulation of people who have the disease initiate in the bulbar region. And so this tool allowed our company to actually jump in at a level where other methods couldn't, because we actually have this essentially complete understanding of what's driving the initiation of ALS from a mechanistic perspective. And just by taking your mouse and circling around any of these groups or people, you can actually make a comparison. And that's what this person's doing here, saying, hey, I want to know what's the difference between this group of people who have limbic initiation and this group is more similar to bulbar. And it tells you exactly, and this is what Jay's going to use to uncover the etiology. And then this is my last slide, and this is, this is something exciting that we've been diving into, is we're marrying digital biology and systems biology with our artificial intelligence. So what we do is we allow the machine to shatter the data set and then to extract causal variants. And then what we do is we actually construct digital perspectives of how drugs or genetic engineering would work at the biological level. And this is now extracting a perspective for us in these diseases that Dr. Anna and the crew are interested in, and figuring out how do we actually attack these diseases. And that's what we're bringing this. So I'm excited to be doing this with Jay. And that's it. And you can look me up on LinkedIn. Thank you.